Friends, welcome to Biology Made Easy. We are discussing enzymes, and today in this video we will discuss the different properties and factors that affect the enzyme action. So first we will discuss what are the different properties of enzymes. Now, number one, what is the nature of one enzyme? Enzyme is a protein, but what type of protein it is? We have classified them as primary, secondary, tertiary and quaternary. So what type of protein? It is usually a tertiary protein, because only the tertiary proteins are three-dimensional structure, so they are functional, and the examples of tertiary proteins are enzymes. So these are tertiary proteins. Again, there is another classification. We have discussed whether the proteins may be simple or conjugate, and in the chemistry we have discussed that most of the enzymes are hollow enzymes. That means along with amino acids there is some non-protein part, that is cofactor is there. So these are also conjugate enzymes or conjugate proteins, you can tell. And the fourth one, again we have discussed that the proteins may be globular or fibrous depending upon the solubility. So all enzymes are soluble in water, so these are globular proteins. That means enzymes are tertiary, conjugate and globular proteins. In one sentence also you can write all those things. Inside this cell, in what form these enzymes are present? They are present in the form of colloids. And these enzymes, they always function only when they are present in very small quantity. The main function of the enzyme is to accelerate the rate of the reaction, but only when we will give in a very small quantity. If we give a very huge amount, then it will inhibit the rate of the reaction. The next point, the enzymes always remain unaltered at the end of the reaction. This is a major difference between enzymes and hormones. Usually, the enzymes at the end of the reaction, they remain same. That means they are not utilized. Their structure is unaltered. But hormones, like enzymes, they aid a chemical reaction, but at the end, they are consumed. This is a major difference. Very often it is asked in the examinations. Then next point is, they accelerate chemical reactions. They accelerate chemical reactions, but do not disturb equilibrium. Do not disturb equilibrium. This is a very important point. Suppose let us take one example. Suppose in one reaction, water and carbon dioxide, they combine to give rise to bicarbonate, and again this bicarbonate, it can dissociate into water and carbon dioxide in presence of enzyme carbonic anhydrase. So this carbonic anhydrase is accelerating the rate of the reaction both in forward and backward direction, but it will never maintain equilibrium. Suppose water and carbon dioxide will combine together to give rise to bicarbonate. Then the same enzyme, when this reaction will take place, when carbon dioxide concentration is high. Now the reverse reaction, the bicarbonate will break into water and carbon dioxide, again the same enzyme carbonic anhydrase. And when this reaction takes place, usually this reaction takes place in the lungs when carbon dioxide concentration is low. So you see, this same enzyme is catalyzing both forward and backward reaction under different conditions, when the carbon dioxide concentration is high or low, but it will never maintain equilibrium. That is one of the very important properties of enzymes. Then next we can discuss the enzymes are always specific in nature, and this specificity of the enzyme always is due to the type of amino acids, or you can tell the amino acid sequence present in the apoenzyme part. That determines its specificity. For example, if we take example of glutamate, pyruvate, transaminase. Now from the name, you can guess what type of function it is carrying out. Transaminase means only it will transfer amino group. Again, from what to what? From glutamic acid or glutamate, which is an amino acid, to pyruvic acid, which is organic acid. That means this enzyme cannot transfer any other group. It cannot transfer phosphate or hydrogen or any other group. It will never transfer. It will only transfer amino group. And again, the name glutamate pyruvate means from glutamate to pyruvic acid it will transfer, not from any other thing. That is, it is a specific nature. Next point. When there are a series of reactions, suppose you take the case of glycolysis in case of respiration, or Krebs cycle, or in case of photosynthesis, you take the case of C3 cycle. There is a series of reactions taking place one after another in a sequential manner, and that is known as a metabolic pathway. So here the enzymes, they will function at a particular place when that particular substrate comes. That is known as a metabolic pathway. Metabolic pathway. That means when there are several reactions takes place in a sequence, the enzyme will react only at the particular place. It will not react in all the reactions. Next point. Each enzyme has one turnover number. Now what is this turnover number? In unit time, per unit time, how many substrates are converted to products? Per second or per minute or per hour, how many substrates are converted to products? So usually the substrates are converted to products only when through the active site. So the more the number of active sites, the greater is the number of substrates that will be converted to products. It depends upon the number of active sites, because sometimes enzymes may have 1, 2, 3, 4 hundreds of active sites. So the turnover number depends upon that. If you take the example, you can take the example of this carbonic anhydrase. The carbonic anhydrase enzyme, it per second it converts 6 lakhs of substrates into products per second, and it is highest among all enzymes. Per second it's converting 6 lakhs of substrates into products, while the slowest one is lysozyme. Lysozyme converts 0.5 per second. That means for converting one substrate into product, it requires 2 seconds, the slowest one. So these are some of the properties of enzymes. Then coming to the different factors that affect the enzyme action, there are 3 main factors. One is temperature, another is pH, and the other is substrate concentration. Now we'll discuss the effect of temperature on enzyme action. Usually all enzymes are thermolabile. Labile means sensitive, thermo means temperature. So they are thermolabile. Enzymes are thermolabile, temperature sensitive. So why they are very sensitive to temperature? Because they are proteins. Now usually in most of the plants they function in temperature range of 25 to 40 degrees centigrade in most of the plants. Particularly in human being, our body temperature is 37 degrees centigrade. So in case of human beings, they usually function between 35 to 40 degrees centigrade. But there are some exceptions. Suppose you take the case of conifers, lichens, which survive in high mountains where there is snowfall. They can, in them the enzymes can operate in even the minus temperature. But there are exceptions. So normally these are the ranges of temperature in which they will function with their optimum capacity. And again you have to remember one point. Several enzymes are present inside the cell. This is the temperature range. Each enzyme has an optimum temperature at which it functions with maximum efficiency. Optimum temperature. Each enzyme has an optimum temperature. Some enzyme may have 25, some may have 30, some may have 35, 37, 40 like that. But it has specific. That is, each enzyme has specific optimal temperature at which it functions with the maximum efficiency. Now, if temperature will decrease and increase, what is happening? If temperature will decrease to 0 degrees centigrade, what will happen? The enzymes will be inactivated. That means the enzymes will remain as such. There will be no difference in the structure, but they will be inactivated. They cannot act on the substrate. This is the reason why we keep the food materials, fruits, vegetables inside a refrigerator. Because inside the refrigerator the temperature is 0 degrees centigrade. So the enzymes which are present in the fruits, vegetables or food material, they will not function. And also in the air, whatever the bacteria are there, or the enzymes they are present in those organisms, they are inactivated. That is why they cannot function, and the fruits, vegetables will remain fresh for a few days. That means they are inactivated. But when the temperature will rise above 40 degrees centigrade, means it will rise to 60 degrees centigrade or more than that, then you know that enzymes are tertiary proteins. The tertiary proteins will be converted to primary proteins, and primary proteins are non-functional. That means in high temperature, the enzymes are denatured. Denatured means their nature is D, is a reverse back. They are converted to primary proteins. Now this is the basic difference. At 0 degrees centigrade, they are inactivated, while at 60 degrees centigrade, they are denatured. This is the major difference. And when the enzymes are denatured at high temperature, that means there will be no enzymatic activity. But here again some exceptions are there. Like if you take hot springs where some bacteria, archaebacteria are there, and you know also Taq polymerase. Taq polymerase obtained from Thermus aquaticus, which is used in polymerase chain reaction. It can tolerate up to 100 degrees centigrade because it lives in hot springs. But those who can survive in high temperature, there are exceptions. Those which can survive in minus temperature, there are exceptions. We are discussing normal ones. So in normal case, if it is coming to 0 degrees centigrade, their enzymes are inactivated, and if they will be above 60 degrees centigrade, they are denatured. This is the reason when we suffer from fever, our temperature increases from 98 Fahrenheit to 102.3. Our body shivers. Why? Because the enzymes, they cannot function properly, because the temperature has risen slightly. And if in a slight rise in temperature, this is happening, our body is shivering, then you think of global warming. At present, in the atmosphere around 360 to 400 carbon dioxide is there. Due to global warming, it will increase to 500, 600, then the atmospheric temperature also increase to 50, 60 degrees centigrade. What will happen to us? The human being along with all other animals will be extinct. That is why the climate change and all other things are going on. So this is the basic difference between the low temperature and high temperature. Now if we will plot a graph from 0 degree centigrade to higher temperature, what is happening? Let us see. Suppose this is temperature 0, 10, 20, 30, 40, 50, 60, 70. This is the temperature in centigrade. Now this is the rate of the reaction. Suppose the temperature is 0 at this point. 0. At 0 degree centigrade they are completely inactivated. Now with a rise in temperature, the rate of the reaction will go on increasing. And to what extent it will increase, it depends upon the optimum temperature. If it may be 30, 35, 40. But we have discussed 40 is the highest one. So up to 40 degrees centigrade it will increase. Then after that it will gradually decline. That means slowly it will increase and then will decline. That means at this point, it is the optimum temperature. Optimum temperature. That means that particular enzyme will function at this, suppose it is 38 or 39, at that temperature it has optimum activity, or it can function with the maximum efficiency. And this optimum temperature is specific for a specific enzyme. But how this rate of the reaction is increasing? For this, we have to know what is temperature coefficient. Temperature coefficient, which is designated as Q10. So what is this Q10 or temperature coefficient? Q10 is equal to rate of reaction at a particular temperature, suppose x plus 10 degree centigrade, divided by rate of reaction at x degree centigrade. That means say at a particular temperature, rate of reaction at a particular temperature divided by rate of the reaction at 10 degree less. This is known as Q10, and this always comes to 2. Suppose we'll take the Q10 value at 30 degree centigrade. What does it mean? The rate of the reaction at 30 degree centigrade divided by the rate of the reaction 20 degree centigrade, and it will always come to 2. So now listen carefully. A general rule of thumb is that for each 10 degree rise or fall in temperature, the rate of the reaction is either doubled or reduced to half. For each 10 degree centigrade rise or fall in temperature, the Q10 value or temperature coefficient usually doubles or reduced to half. This is the general rule of thumb. Suppose the temperature rising 10, suppose the temperature is at 25 degrees centigrade. If it will be the temperature will rise to 35 degrees centigrade, then the rate of the reaction will be doubled. For each 10 degree rise, it doubles. And suppose the temperature is at 30 degrees centigrade, at 20 degrees centigrade, what will happen? It will be reduced to half. because for each 10 degree rise it doubles, and for each 10 degree it becomes half. That is why it always gives a value of Q or the temperature coefficient is equal to 2. So this is how the temperature affects the rate of the reaction. And this type of curve you can call it as parabolic curve. Parabolic curve means it gradually increases, then it declines. So this is how the temperature affects the rate of the reaction, and how the pH and the substrate concentration they affect, that we'll discuss in the next video. So thank you very much for watching it. Again, we'll meet in the next video. Have a good day.